So today I'm going to talk about predicting population performance based on population's relatives and its neighbours. And so this is important because very often in demography and population modelling, what we do is we measure a few populations in an area, and then we sort of implicitly assume that all the other populations of that same species behave in that area behave the same way or in that general region. And we don't really know if this is true. And so we tested this using a global database of population matrix models for plants called Compadre. And this is a database with thousands of populations and hundreds of species. And so we looked across species and across different regions across the globe. And what the aspect of demography, so demography first is the study of how survival, growth, and births, fecundity, influence population growth rate and population dynamics. And the aspect of population dynamics we were looking at, and the aspect of demography we were looking at, is called elasticities. And elasticities are basically how important those vital rates are to population growth rate. And so we looked across many different species, and all these species have matrices that are different sizes and different forms, and we need to standardize them some way. And so we used a property of elasticities that they all sum to 1 to help us do this. So what we did is in any matrix, you have fecundity transitions, stasis transitions, where a plant stays the same size, growth transitions, where a plant gets bigger, and retrogression transitions, where a plant actually shrinks and gets smaller. And because within a matrix these all sum to 1, you can sum all of these different types of elasticities together, and it still tells you something sensible about how that population was behaving. And so we did this for every matrix, and we produced a vector of 4 elasticities, which were progression, so growth, stasis, shrinking, so retrogression, and fecundity. And we know these aren't independent because they all sum to 1. So if, for example, fecundity elasticity is very high, we know the other 3 must be very low. So to get around this, we put them into a PCA. And what a PCA does is it looks for sort of correlations between variables, and it tries to find the best, rotate them so they have the best axes. That explains most of the variation. And we can see our PCA did a pretty good job. So the first axis of the PCA explains 60% of the variation of the data. And at one end of this axis, we have populations for which stasis is very important, but fecundity and growth are not very important. And then at the other end of the axis, we have populations for which stasis is not very important, but fecundity and growth are very important. And we see retrogression here goes straight up, and that basically tells us that that's not correlated with any of these other things. And so what we're trying to predict in this analysis is a population's position on that first axis. So really this analysis is telling us about stasis, growth, and fecundity. And so what are we predicting them with? Well, we use these exponential decay models. And what these tell us is basically if we have our target population, and then we have all the other populations that are a certain distance from that, we know the distance between the populations, then we can fit this sort of negative exponential decay model where we use this alpha parameter to determine how quickly the predictive support from populations decays. And we can do that with distance because we know the physical distance between populations, and we can do it with phylogeny because we have these species in a phylogeny. And so if we have our target population, we know how closely related all the other populations are in the database are to it, and we measure that with a thing called millions of years since the last common ancestor. So it's basically since the amount of time since they diverged. And so we put this into a statistical model, and we see how the statistical model works, and we can see here it works pretty well. So along the bottom here we have our observed position along that PC1 axis of the PCA, and up the side on the y-axis we have the prediction by our model. And we can see the model actually does pretty well. So the red line, if dots on the red line are really close to the model did really well for. So if all the dots were on the red line, our model would be really good. If hardly any of the dots were on the red line, our model would be very poor. We can see our model actually does pretty well. It predicts pretty well for lots of populations. There's a few populations where it doesn't work so well. And so if we now look at a measure of model performance with R squared, we see that when we predict from all of the populations, which is this pink distribution here, we do really well. We have an R squared of above 65%, which is really good for ecology. But we have in our database, we have a lot of populations which are of the same species in exactly the same place. And these aren't very helpful. If we have a population already of the same species in the same place, well, of course we know what it does. And also these very often, if this is the case, these populations are all from the same study. So they're also done over the same time period, by the same people generally, and so they're definitely not independent. So we want to see how much of our predictive power has been explained by that sort of thing, and how much has just been explained by the distance and the phylogeny. And so we can remove these self-prediction, these selfies from the database that we're using to predict each population. So when we run the model, we just take out those populations and we use the rest of the populations to make the prediction. And we can see when we do that, the R squared falls off quite a lot. So it goes from about 65 down to about 25. So we're explaining 25% of variance in the data now. That's still pretty good for ecology. So it seems as though we do have some predictive power. Populations do tend to act somewhat like their neighbours, and they tend to act somewhat like their relatives. And now if we can remove even more data, so we can say just how much of this is being predicted by close relatives or any relatives. And we can see that it's about 12.5% of an R squared, about 12.5%, which is not too bad. And if we ask how much is being predicted by spatial location, so by near neighbours, not in exactly the same place, but still just nearby, we can see that it's actually getting up above 20% of R squared. So that's pretty good. And now if we see below that, we just have the spatial decay parameters. So these are the actual models of support. So this is how much support a given population adds to that prediction. And we can see that populations that are within about 20 kilometres of a target population are providing pretty much all the support. Once you get past 20 kilometres, a population is providing pretty much no support at all to that prediction. And so this means we need other populations close by can really only be extrapolated within a very local area. For species, we see this as actually we have a bit more, we can extrapolate a bit further, even though the R squared based only on phylogeny is a bit lower. We see for species that it's really within the last, so divergence since 50 million years ago, you're still getting support from species that diverged that long ago. So for reference, the grasses diverged somewhere around 40 to 50 million years ago. So this is sort of saying things like everything in the grass sort of group act in a similar-ish way. So we have some predictive power at that level. So it seems we can extrapolate some demography, at least the elasticities of it, but we can only do it at quite small scales. So we can only do it at sort of less than 20 kilometre scales, and we can only do it between relatively closely related species, so divergence since about 50 million years ago.